So in this session, you're going to look at the principles of biological drawing and then how to make plan drawing and detailed drawing in biology. Okay, so these are the components or the parts of a microscope. So I hope that when you do the exercise, you will be able to fill in the blanks and you can check the answers from here. Now, first thing is when you want to make any biological drawing, you need to use sharp HB pencil to do your drawing. So therefore, please check your pencil case now and see whether you have got HB pencil or not. If you don't have, please go and buy one. Nowadays, you can buy online, right? So you need to have got sharp HB pencil. HB, you know, is lighter than 2B. So when you draw onto the paper, any biological drawing that you use HB to draw, it looks more tidy. And when you erase, it's easier to erase if there's any mistake. So you need to use a sharp HB pencil. If you don't have, go and buy. If you use mechanical pencil, also can. Make sure that the lead that you use is a HB lead. Okay, please go and buy the HB one, or you can share among yourself. Okay. In the next practical, when you come, the first practical you do is actually biological drawing. So make sure you remember to bring a sharp HB pencil. I will check. If you don't have any sharp HB pencil, you cannot enter my lab until you go and find one for yourself. Okay, so you better buy now and get ready now. No sharp HB pencil, no entry to the lab, right? So when you make your outline or you draw, the line must be sharp. The drawing line must be sharp. It must be clean, single line, and it must be clear. For example, here. So this one here, you see the line is clean, single line, clear, and the thickness is the same throughout. Whereas compared to this one, you can see that there's some feathery line here and some feathery line here. The thickness is not the same. So this is not acceptable. Or here, feathery and gaps. Especially when you draw circle, when you draw an organelle like this, right? So you need to draw in a way that you don't draw it like this or like this. Okay, you can't find these two objects in the cell one. If you draw like this, means we have got a new discovery, a new type of parasite that live inside the cell. So we're going to examine what are these two things. So circle means you must draw a complete circle without a gap or without feathery line. And also no shading. All biological drawing must not have any shading, whether you shade the whole thing or use lines, you use dot, you use pattern, cannot. They are all not acceptable. Then what is biological drawing? All right, you see here, this is a photograph taken under the microscope, so it's a micrograph. If you ask any artist to draw, they can repeat the whole thing, but they actually do not know what they're drawing. So if you're a biologist, when you draw, you need to, number one, identify the biological structure that you can see in that micrograph, and you draw them, right? Like you know that this is nucleus, and this nucleus got two membranes that you can see, and there is heterochromatin, there's nucleolus, okay, and there's this membrane-bound organelle, which looks like ER, and there are mitochondria here. So these are the structures you need to identify and to draw them. And after you identify and draw them, you see this drawing, right, can exactly superimpose with the micrograph exactly. So this is called a good biological drawing. It must be very accurate in terms of the shape, the location of different structures in object, the size. So if your drawing is accurate, you will be able to superimpose your drawing exactly to the image, exactly to the image that has been magnified to the same size as your drawing. So that's called a good biological drawing. And of course, you cannot draw a textbook drawing like this. You just memorize from textbooks and then everything out during the exam. Cannot, no mark for this one, definitely. Okay, next. Sometimes they will ask you to draw 3 cells. So you need to choose the best 3 cells to draw. If they ask you to draw 3 cells, say for example, you use the microscope and look into the slide and you can see this thing here. And the question asks you to draw 3 cells. So you cannot just simply choose 3 cells to draw one. You need to look around the specimen to choose for the 3 cells where the structures inside the cells are quite clear for you to see and to draw. Then you draw them. Okay, so you need to choose. And then relative proportions should be correctly represented. So you look at the drawing here. Now this one represents these 3 cells. These 3 cells, right? Can you see the drawing is so accurate? It's quite accurate in terms of the location of the nucleus, the size of the nucleus, the shape of the nucleus, the length of the cell, the length of the cilia. So a good drawing is your drawing must be able to superimpose with the original micrograph of the same size. They will superimpose exactly. That's called the good drawing. Now, in the nucleus, you can see nucleolus as well as the heterochromatin. So therefore, you need to draw them. You actually need to draw them because you've learned those structures. So can you see that inside the nucleus is not empty? There's something there, but there's no shading because you cannot shade. Cannot shade means you cannot put the dot also because you use dot for shading purpose. You can see a dot here, but you cannot dot there. You can only outline an object that's big enough for you to outline, like this one here. Okay, so I just to illustrate, let's say you see this one here is represented by this one here. Can you see or not? This one is represented by this one, right? This one is represented by this one here. Can you see or not? And even the side here, can you see the side here is represented by this drawing here. So this is called accurate biological drawing. So this is the accuracy that I want all of you to achieve. Right? Now, a space will be given to you in the question paper to make your drawing in the exam. You must make sure that your drawing must occupy more than half the space provided for you. Best still, 3/4 of the space given for you. The bigger, the better, because the bigger means you're able to draw the detail quite clearly for the examiner to see. So I suggest 3/4 is the best. Half is also too small. 3/4 of the space given, and then leave enough space on one side for you to do your labeling. So this is a space here which you can use to do your labeling. And then if you're in the lab, then you leave the space for you to write the title. In exam time, you don't have to write the title because you may not know what specimen it is. So therefore, you don't know what is the title. But in the lab, you will know what is a specimen. Therefore, you have to write the title. So what do you mean by writing the title? See, can you see the title here? A title must start with a figure number. So this is the first drawing that you draw in the lab. So figure 1, then followed by figure 2, figure 3, figure 4. And what exactly are you drawing? You're drawing 3 ciliated epithelial cells, right? But all animal cells also got ciliated epithelial cells. So which animal is from? Okay, it's from rabbit. But rabbit got so many organs. A lot of organs also got the ciliated epithelial cells. Lung also got. Oviduct also got. Which one? Oviduct. And oviduct is 3-dimensional. Which section are you getting from the oviduct? So transverse section. I'll talk about section later on. So this is a full label. So after you, full title. After you draw the whole thing, remember to write a full title there so that in future, when you look back into drawing, you know exactly what this specimen is that you have drawn. Very important. Okay. Next. Now, when you're asked to label, please also use pencil. Everything in pencil. Drawing in pencil, label line in pencil, label, annotation, everything in pencil. Okay. Now, label line, please use horizontal line that you draw with ruler. So it's ruled line or vertical line. So I only accept horizontal and vertical line. I don't accept any other lines. Okay. And please label systematically from top to bottom, from outermost to innermost, as much as you can. Systematically do that. Okay. Top to bottom, you know lah. Outer to inner means, okay, cell surface membrane, cytoplasm, nuclear envelope, and then this is heterochromatin, nucleolus, cytoplasm. Can you see that? Okay. Wait. Okay. Now, label and annotation is different. Label means you give me the name of the structure. Annotation means please describe about the structure that you can see. So describe, you can tell me about the shape. You can tell me about the size, the color, the texture. Texture means are there a lot of dots in it? So it's granulated. Or is it like smooth, plain? Okay. So this is called texture. Smooth looking or granulated. Okay. Now this is the title. Now you see in the title, I don't write magnification for my drawing. My question is that why is it I cannot write magnification for my drawing? If I can calculate magnification, I can draw, right? If I don't calculate, I cannot draw. But some students come and tell me, but teacher, the microscope, right, is 10 times 40 I used to draw my cell. So therefore, why not I put that 10 times 40? My microscope is 10 times 40, right? So I said, no. Januli, can you tell me why I cannot put 10 times 40 for my drawing?
I'm not sure.
Okay, never mind. Someone else try. Philbert, would you like to try?
Is it because it's not the actual image, but it's just a drawing you drawn by yourself?
Correct. Yeah, imagine, I take the slide. When I use my naked eye to look at the slide, that's the actual size of the specimen, right? Okay, let's say I got a stem there. The diameter of the stem on the slide is the actual stem diameter, right? Then I put into the microscope and I view under the microscope at 10 times 40. That means whatever image that I look into the microscope, that image size is 10 times 40 is correct. But when I look into the microscope and I draw it onto my paper, do you think your drawing is same size as the image or bigger? Hello? Is your drawing same size as image or bigger?
Bigger.
Yes, usually bigger, right? So it's bigger. Can it be 10 times 40?
No.
Cannot, isn't it? So that's why your drawing size is different from the image that you see immediately in the microscope. So therefore, you cannot write 10 times 40 because drawing size and image size is different. Unless you take a photograph of the image and then to the photograph, you label the magnification 10 times 40. That was correct. Because that image is taken at 10 times 40 in the microscope, but your drawing is not. So that's why never write the magnification of your microscope onto your drawing, because your drawing is not the image in the microscope. Okay? So you need to understand clearly why you cannot do something, right? When you understand, you will not repeat. When you don't understand, you repeat. So therefore, biology, you need to understand every single thing that I teach you, you need to understand. When you understand, biology becomes very crystal clear and very easy. It is very easy. So your focus is understand. Okay, right. So I've talked about label must use ruler to draw the horizontal and vertical line, and it must point into the structure. That means your label line must end up inside the structure that you want to label. Cannot end up at the border. Must be point into the structure, like nucleus point into the nucleus. Okay, cell wall point to the cell wall. Unless you cannot point into because it's just one line, like cell surface membrane, one line. So just point to that line, or else must point into the structure. There's a space there. Do not end up with arrow or dots. It must be plain single line. And the line must not cross each other. So label line do not cross each other. That's why I ask you to use horizontal line. If you start to use slanted line, you start to see crossing here and there, very messy. Cannot. Annotation is an additional notes, right? Usually we write it as a bracket behind the label. Title, please write it when you do your drawing in the lab, but you don't have to write title in the exam because you do not know what's the specimen, so you cannot write the title. Okay, so title must start with figure number, name of the structure or cell or tissue organ, name of the organism of origin, and don't forget the viewpoint. Remember just now the TS, LS, OS? So let's say this is a piece of carrot, right? You make a section through it. A thin slice means a section through it. So this is transverse section. Hello. This is transverse section. This is longitudinal section. So transverse means what? Transverse is cross sections, like this is the road. You cross the road, so you go across the road. So you cut through this way. So this is called cross section or transverse section. All right. Longitudinal section is you cut along the length of the object. So there's just longitudinal section, like this one here, cut along the length of the carrot. This is oblique section, means you cut slanted along the length, not along, slanted with respect to the axis of the length. So this is called oblique section. So oblique section usually is oval. Okay, cross section is round, and longitudinal section usually is rectangular. Now this example of a drawing and the labeling and the annotation. So in the past, usually when you do your own labeling, we use all capital letter, right? This is what I'm being taught in university. Use capital letter to do the labeling, and then annotation as bracket, right? But in the exam time, you will not be asked to do that because you don't have time. They only test you. Later on, I show you what you mean by that. Now, this diagram, everything is okay except that the label line ends with a dot. This is wrong. It must be a single plain line, or end up with an arrow. This is wrong. It must be a single plain line, right? Other than that, the rest are correct. Now, this is a left side is a micrograph of the root, and the right side is a drawing of a student. So you need to look at the drawing and tell me what are the errors of the drawing that you can identify from here. So look at it, and I'll call you. Please tell me what is the error, okay? First one. Curti. First error in the drawing.
It's not complete.
Not complete, yes. You can see the structure on the left side and the right side. The drawing has got less things that you can see, right? So not complete, yes. Number two, Shumin.
Not clear.
Not clear. Which part is not clear? You mean this part is not clear? This part is not clear. This part is not clear. Worst is this one here. You see this one? You see this or not? There's a gap there, right? Okay. Number three, Samir.
There's shading.
Okay.
There's shading on the drawing.
Yes, shading. Shading is not allowed in biological drawing. It's not necessary. That is very messy. All right, correct. Generally,
The label lines are slanted and not horizontal.
okay. Now, because the student used slanted label line, you start to see the two label lines cross each other, right? See here, so it becomes very messy. And didn't use ruler, so that's why it's very messy. All biological drawing, you cannot use ruler. All label line, you must use ruler. So you can distinguish very clearly the straight line that you see, is it a label line or is it the line of the biological drawing? That's why biological drawing, even if it's a straight line, you must use freehand to draw the straight line, like cell wall. But teacher is very straight one. Yeah, but you cannot use ruler to draw biological drawing. But label must use ruler. Sorry. Ruler doesn't look like this. Ruler look like this. Straight, straight. Okay? So that will distinguish between part of the structure that you draw versus your label line. So there's a reason behind why the rules like this one, not simply one. So if you understand, then you know why. Okay? Can you see that the title is not accurate? Root structure. What do you mean by root structure? Root of what plant, right? And then is it TS, LS, or OS? So all these things must be written. And I also suggest that the drawing needs to be bigger a bit, right? This is about half the size of the space given. So it's still very small. So at least draw 3/4 the size. Then the size is almost okay. Cannot draw too big. When you draw too big, after you got not enough space to write your label, and some students even draw into the words of the question. The moment you draw to the words of question, you lose 1 mark. Okay, so you need to plan the size of the drawing. So this is a question given to you as an exercise. Please do it in your exercise book. Okay. Now, a good drawing after I draw, right? It must be able to superimpose exactly with the micrograph. If it's a good drawing, it must be able to superimpose. Here, nothing can superimpose, so you know it's not good drawing. Okay, right. So this one is to show you if you see 2 images of an object, one is TS, one is LS, you will be able to guess what is the possible 3D shape of the image. Now, because when you see anything under the microscope, they are all flat one because you have to make it flat for the light to pass through. So therefore, you can only see 2D, you cannot see 3D. So in order to figure out what is the actual 3D shape of this particular structure that you see in microscope, you must look at the TS and LS image to to predict or to figure out what is the 3D shape. Right, so TS is like this, LS is like this, 3D shape must be spherical. Right, TS is like this, round, and LS is like this, rectangular. So therefore, probable 3D shape must be a cylinder. Right, so from TS is this, LS is this, so this is a cigar. Right, this is a cigar shape. Right, 3-dimensional. So this is something that you need to know. Actual object is 3D, but what you see under microscope is 2D. Then can you figure out what is 3D? This kind of question came up in exam before, but very rarely, but came up before. Okay. Now you can play around with this one. You go and play my PowerPoint when you do revision, and you can play around with this question, right? Okay, so there are 2 types of biological drawing. What I'll teach you today is only the detailed drawing. Okay, I will not teach you plan drawing. So first, there are 2 types of biological drawing that you need to learn in your syllabus. The first one is plan drawing, and the second one is detailed drawing. Today, I will not talk about plan drawing. I will talk about detailed drawing only. Right? So let's look at the detailed drawing. For detailed drawing means you draw the cell and all the visible biological structures of the cell that you have learned. So that's called detailed drawing. So we're not going to look at plan drawing yet. Draw the cell and all the visible biological structures that you've learned that you can find inside the cell as well as outside the cell. You know that that is a biological structure, so you have to draw in detailed drawing. So detailed drawing, you focus on the cell and the structures. Okay. Okay, here, there you go. So again, example, which I've shown you just now, the question asks a student to draw 3 adjacent cells. Adjacent means adjoining, means they're side by side joining each other. So choose the best 3 cells and then you draw them. Okay, so this is the best 3 cells you choose and you draw them. Now, if sometimes instead of giving you the slide, they may give you a micrograph in the exam paper for you to make the drawing. So example, I give you this micrograph in the exam paper and ask you to make a drawing of the ciliated epithelial cell. Let's say 3 ciliated epithelial cells. Now, in this drawing, I cannot see individual cilia, right? Individual cilium, I cannot see. I think the cilia is, this is the brush border, okay? But not very clear, you can see or not? This is the brush border of the cell, not very clear, right? So therefore, the only thing I can see here is this shadow, but I cannot see individual cilium. Because of that, I will draw this. If I can see individual cilium, I have to draw this. So it depends what you can see in the micrograph. But for microscope, it is your duty to focus as sharp as possible, then you draw. So usually when you focus it very sharp, you should be able to see cilia. If you cannot see cilia, there is something wrong with the focusing already. So next time when you go to the lab, please learn up how to use a microscope so that you know what are the things that you can adjust to make the image the sharpest possible for you to make your drawing. So that one you have to learn in the lab. Okay, no play. No say that, oh, I let my friend do all the focusing for me and my friend will make the drawing. I just copy the drawing. Easy peasy. Yeah, easy peasy now, then you die in exam time. You struggle in exam time. Huh? So don't do that. All right. You must choose wisely what to do and what not to do. Okay. Now this is example passive question and the student's answer. So the students asked to draw 3 epidermal cells and 3 of the palisade cells touching this epidermal cell. So this leaf, right? So 3 epidermal cells and 3 palisade cells that are touching these 3 epidermal cells. So you need to only draw what the question asks you to draw. Okay? And you must choose the best cells to draw. So the best cells that you can see in the slide is very clear. Everything is very clear. You can see clearly. Then you choose those cells and draw. So you have to scan around the slide and look for area that's very clear and draw them. And every time you draw plant cell, you need to draw middle lamella, which is this one here. Then you say, teacher, I cannot see the middle lamella. You cannot see because you have not focused it until the sharpest possible using microscope. So you have to learn how to do that. Okay. Then you see this question here. On your drawing, use a label line and a label to show one palisade cell. So this is a label line which is ruled. That means you use ruler to draw, and then you label the palisade cell. So the student decided to label palisade mesophyll cell, which is actually correct. If you don't write mesophyll, also is correct because the question asks you to label palisade cell, right? But it's actually palisade mesophyll cell. So you only label what you're asked to label in the question. Don't go and capo and label everything and then everything correct. Also, you don't get marked. What's the point? Right? You must well spend the time to do experiment and get your results correct, right? Okay, so you need to know how to use your time efficiently. What is important, what is not important. Okay, that's called smart. You study smart and you answer smartly. This is called smart. But smart doesn't mean lazy. You cannot lazy. You can be lazy smartly, but you cannot be smartly lazy. Which one is correct? Lazy smartly or smartly lazy? Okay. Now, which one is correct? A, B, C? Joyce?
Yes, teacher.
Which one is correct?
C.
Why A is wrong?
Without labeling.
Okay, right. Now, your answer is correct, because you said no labeling. Actually, I cut the labeling away because I got no space to put my labeling. These are all 3 drawings of 3 different students of the same image that I project onto the whiteboard for them to draw. How come same image 3 students draw different thing one? Can you see or not? This is the best answer. You see, if a hand got so many fingers, right? How come these students suddenly from so many fingers become 4 fingers? You eat away other fingers already lah. Okay, so this is the most accurate because you draw as detailed as you can see. This one, I don't know what happened. Maybe the student has got short-sightedness and didn't wear glasses, so sit behind the lab, and then from 10 fingers becomes 4 fingers. Okay, next, B is wrong because of shading. Now, this one is drawn by examiner who came to Taylor's College and give training to the teachers. The examiner draw one, so I took a photo of the whiteboard, on the whiteboard that she drew. Now, she illustrates different ways of labeling by the students that you can see in students' answer in exam paper. A is wrong. Can you see or not? Because one gut cell, how come the student label gut cell? So A is wrong. B is correct. Okay, but it's the best to point into the cell. B is wrong. B is wrong because label line must point into the cell. C is correct. Okay, it's pointing into the cell. D is wrong. You can use bracket to label a structure, but the bracket must touch the object that you want to label. In this case, the bracket is touching the space outside the cell, so it's wrong. So this is correct way. If you want to use bracket, it must touch the object. And this one is also wrong because when you label 2 cells, how come your label is for 1 cell? So you must have an S here. So these are all the common mistakes among the students in the exam answers. You have to pay attention to. Again, tree drawing, which one is correct? I think between A, B, C, C is more correct because there's a limited lamella. But I think A and B are more accurate in terms of the proportionality. That means when the 3 cells are long and thin, how come in C, the long and thin become short and fat? So in that case, A and B, the shape is more correct, but C is more correct because there's middle lamella. All right. So that's all for today. We have completed the lesson for making a detailed biological drawing. All right, so I will let you know whether I want you to do an exercise on detailed biological drawing in your Telegram. All right, so meanwhile, do all the questions and exercise I ask you to do and make the reading. Pass up next week, Monday lah. Next week Tuesday, during practical session, I will check your answers. Okay? All right. So do you have any question?